Hi, everyone, and welcome. Thanks for joining again. If you were here last night, especially, glad we can spend our weekend together. I see we already have a good amount of questions, and I've actually made some slides accordingly as you guys were posting them. So I'm going to share my PowerPoint really quick. I can share the link with you as well. Oh, I may actually need to change the editing settings real quick. It's mainly just pictures at this point. That's like kind of how I function. If you guys have listened to my sessions before, you know that. I don't really have any words on the slides because I talk the words. But I definitely have some things up there about what all did I put in there. CRISPR gene expression, CAM plants, etc. So I'm going to get started right away. So the main focus of today is going to be big ideas 3 and 4. So some examples. Ooh, I don't know why that happened. Some examples of things that we can talk about today: DNA and RNA, cell cycle, mitosis, meiosis, all sorts of genetic stuff, gene regulations, genotypes, phenotypes, cell communication, signal transduction, the nervous system, biological molecules, cellular organelles, organ systems, communities, ecosystems, enzymes, cooperative interactions, populations, cellular variations, genotype expression, biodiversity. Wow, those are just a few of the things that we can talk about, but I would love to answer all of your questions as well. So I have a slide on CRISPR. I have a slide on the various photosyntheses. I have a slide on cyclins and CDKs. I'm going to start answering a few of these other questions that are really, really short, and then I'll dive into those, and obviously you guys can keep asking questions. Do you need to know the functional groups? Absolutely not. Good news. It's like an organic chemistry thing, so you don't even really need to know about them in AP chemistry. So you do not need to know those. No. Endocrine, I will cover in a little bit more detail. Should you know the nutrient cycles? I would suggest knowing a little bit about the nutrient cycles, just like major sources of those things and how we cycle them through. I mean, carbon cycle is just knowing cellular respiration and photosynthesis, which you definitely need to know anyway. Nitrogen cycle, you can just know that there are nitrogen fixing bacteria. Phosphorus is a really easy one because it basically just is erosion that's getting phosphorus back into and decaying. Animals is getting back into it. So is it a big part of the exam? No, but I think it's an easy one to just kind of brush up on just in case it shows up. You also have to realize like some of these I can't answer if they're not going to show up on the exam because I just don't know. You'll know before I do because I never get to see the multiple choice and I don't get to see the free response until Wednesday. Should you know gene mapping? Another great question. I do have a slide in here about it. It's not usually a large part of the AP exam either, but I can just go over some very basics with you so that you feel slightly more confident on gene mapping. But I would not stress overly about that at all. Basics of gene expression, I do have a slide about that. Molecules that can and cannot pass through the membrane. I can answer this one really, really quickly. Anything that's charged is not able to get through the membrane, and that's really, really important to know. So sodium, potassium, right, like hydrogen ions, all of those things, it's really, really important to know they can't get through the membrane on their own because we use that to establish concentration gradients. Things that can get through are nonpolar and small, which it looks like Anusha answered. So like carbon dioxide and oxygen can get through, but anything that's polar or charged or bulky cannot get through because of those lipid tails. Steroids and lipids, they can get through because of those lipid tails, but pretty much everything else cannot. Are plants a big topic on the exam? Absolutely not. They're actually eliminating plants entirely next year. Plants are a very, very small portion. Truly, they only show up when talking about photosynthesis. And there's that one experiment that I'm sure you've all seen about phototropism where the plants are bending towards the light. They own that diagram, so that shows up on occasion, but that's a real easy one. So not a big part of the exam at all. Phylogenetic tree and cladogram are essentially the same thing, except phylogenetic trees, the lengths of the branches are actually indicative of the amount of time. So if you saw free response question number 1 from 2018, that was a phylogenetic tree that they were showing you with the bear populations. But then they constructed a cladogram where the branches sizes don't matter except for where the joining, they don't matter as far as time. They just matter as far as relatedness, but both of those are going to show you how closely related two things are and how recent their most recent common ancestor is. Okay, Cool. All right. I'm going to just start cranking then. Wow. Lots and lots of questions. Okay. Good stuff though. Gene linkage. Okay. I can definitely talk to you about sodium potassium pump. I would highly suggest. So my past 2 reviews, I've gone through that. So either of my past 2 reviews, just because I've gone over it twice already, I probably won't go through that tonight, just because there are other things. Okay. So it looks like CRISPR is of the, well, let me just go through here. So someone asked about, let me just pull this up and say that I'm answering your question. C3, C4, and CAM plants. So I haven't seen this in a long while. And again, plants aren't super, super big on the exam anymore, but it is about photosynthesis. So they sometimes do talk about it. Basically, what you need to know is that C4 and CAM are both adaptations so that plants can save water and not get dehydrated and dried out. So these are going to be both taking place in types of plants that live in arid, dry areas. So with C4 plants, what you'll see is that they fix carbon in a different part of the cell than where they go through the Calvin cycle. And that's so that they can kind of close their stomata. So they can kind of pack them full of carbon dioxide and fix that carbon. You can see it better in actually the next picture. So you can see this here. So our carbon dioxide is coming in, and we can actually then close the stomata and fix the carbon in here, and then go into the Calvin cycle down here, as opposed to in a C3 plant where we just keep these open, and that's where we're fixing carbon. And so we need that open supply as opposed to kind of doing this in a 2-step process so that we can most likely close those stomata more frequently, so that we're not losing as much water, which you can see is going through those stomata in both of these diagrams. So that's your C4 plant. So it's just a different location for carbon dioxide fixation. And then CAM plants are really, really cool because they divide it by time of day. So they only fix carbon at night because that's when the stomata can open and there's going to be less evaporation, less loss of water because the sun's not out. And then they go through the Calvin cycle during the day. So they can actually keep their stomata closed during the day. So they're not getting any CO2 in, but they don't need to because they've fixed all their carbon at night. So that's just a brief overview. Kind of the differences there. So that's all evolutionarily beneficial to plants so that they're not losing as much water through their stomata. C3 plants, which are our normal ones, just kind of allow this process to happen as is. And they're not super concerned about losing water because they usually live in areas where water is not going to be like a super limiting reagent. Hopefully that helps answer that question. Okay. Next, I think I have a little bit with gene mapping. So wherever my gene mapping question was, don't remember. Just trying to make sure that I get through all of these. Now my computer doesn't want to work anymore. Ooh, maybe I already went through it. Not sure. Anyway, gene mapping, gene linkage, whoever asked that, wherever that question is, maybe I just am losing my mind. Basically, what you need to know is that the amount of times that 2 genes separate is indicative of how close they are on the chromosome. So gene mapping is only, and gene linkage is only if 2 genes are on the same chromosome. Otherwise, they go through independent assortment, which means there's a 50% chance that they're going to get separated and a 50% chance that they're going to stay together due to independent assortment. So again, this is only used for genes that are on the same chromosome, and the amount of times that they actually don't end up in the offspring together is called the recombination factor. So here you can see between genes A and B, 13.2% of the time they actually get split up from one another. We can tell that from the offspring, whereas B and C, only 6.4% of the time they're not staying together, and that's because they are much more closely located to one another on the chromosome. So when crossing over occurs, so what you need to think about here is, like, if I'm randomly going to slice this chromosome, there's a much higher chance that it's going to be between A and B than that it's going to be between B and C, just because this is a smaller distance. And so the random nature is going to make it actually less likely that I'm going to cut those 2 apart as opposed to these 2. Because of that, crossing over doesn't usually happen between B and C, again, just because the chances of getting a cut between those 2 is pretty small, which is why they're sticking together like we would expect them to, because they're on the same chromosome. So this is really telling us the frequency of crossing over between 2 genes on the same chromosome. And again, the higher the number, the further apart they are, because it's more likely that crossing over is going to occur because there's more distance that could be cut during crossing over. So that's truly the basics of it. Honestly, it never really shows up but I could be totally wrong and it could show up this time. Regardless, usually they would give you the numbers and you would just need to analyze them. So it's not as complicated as you might think. Okay. Let's see, I think next I have cyclins and CDKs. Okay, so cyclins and CDKs. So CDK stands for cyclin-dependent kinase. A kinase is an enzyme that phosphorylates things. Let me grab my water really quick. My voice is like totally gone from review season. Okay, so CDK, again, cyclin-dependent kinase. Kinase enzyme that phosphorylates things, but it depends on cyclin to do so. So when cyclin binds to a CDK, CDK is then able to phosphorylate targets. Phosphorylate different targets, and when they do that, you can see the little phosphate group here. When they do that, then that's able to activate, in this case, S phase. So oftentimes cyclins and cyclin-dependent kinases are used to regulate the cell cycle, which is why they're really important, because if something's wrong with them, then we could have things like cancer. So here, CDKs actually need the presence of that cyclin, which means that the cell has made it through the checkpoint saying like, yep, we're good to go and start replicating the DNA. When those two bind together, we phosphorylate a couple different targets that are going to initiate S phase. So S phase is where DNA is replicated. So all of those enzymes that are responsible for DNA replication, like DNA polymerase, helicase, single-stranded binding proteins, topoisomerase, all of those things would be activated so that we can go ahead and replicate the DNA and continue on with cell division. So if there's something wrong with one of these processes, like this is overly active, then we're going to see S phase beginning even when maybe it shouldn't, and our cells shouldn't be replicating, and that can lead to cancers. So oftentimes with the cell cycle and things like that, more often than you're going to be asked about why something's happening is like what happens if it goes wrong. And so that would be a really good example there. If this is activated too frequently, we would see overproduction of cells that shouldn't be overly produced. With things like this, you do not need to memorize like what a CDK and a cyclin is. More often than not, they would show you a diagram and they would show you what happens and what activates what, and you would need to determine if something went wrong, what would need to go wrong in order to produce a tumor, or what would need to not go wrong in order to not produce a tumor. If that makes you feel any better. Yeah, you don't necessarily need to have those words memorized. Okay, I think CRISPR is next. All right, CRISPR. So very interesting that someone said, I was going to say that usually something like CRISPR wouldn't show up on the AP exam, but I may stand corrected, as it may be a free response question. So what do you need to know about CRISPR? So oftentimes, one reason why this may show up is that oftentimes College Board likes to tie something in that's very current, and CRISPR is obviously something that's very current. So CRISPR is just a gene editing tool. So I can see them putting this in a free response and thinking that perhaps you're going to need to apply like some sort of other gene editing situation, or talk about transcription or translation, or how this would have an effect on a cell, which is why I have our next slide is going to talk about gene expression, because that was a question as well. So what you can see happening here is that basically we're just editing a gene. And so what CRISPR does, it's going to be an enzyme, a protein that can come in and find a specific region and actually make a cut in that region so that we can input some sort of programmed DNA programmed DNA into this cell. So why is this such a hot topic? So if you've heard of genetically modified organisms, CRISPR is usually the enzyme behind those, because we can go into the DNA, we can slice it open at a particular region, we can input a new piece of DNA. This is very similar to if you were in my biotechnology lecture when we talked about restriction enzymes. So this is just a fancy restriction enzyme that's able to cut in a particular region and then cut a piece of target DNA in that same region and then input it into that cell so that that cell is now capable of making a protein. If this was on a free response, you would definitely want to mention that all this is doing is providing a code so that this cell can now make a new protein. They really, really want to make sure that you understand the trajectory of how things work here with like yes, we've just added new DNA, but what we've really done is given them a program to make a protein, because the protein is the thing in the cell that actually does stuff for us. Our DNA doesn't actually do anything besides code for the proteins that do things. So again, this is just an enzyme that can come in. It's very, very common in genetically modified organisms. It's going to cut a particular target region. The interesting thing here is that it's going to cut, you're going to use the same enzyme to cut the target region as you are to cut the target gene, so that the ends match up. We call those sticky ends, so that it's kind of like a puzzle piece. Like you're using the same puzzle piece or the same like fancy scissors. If you remember those scissors from growing up that were like shaped different things, right? You're using the same scissors to cut the target gene as you are the target DNA, so that when those two come together, they can snap into place like a puzzle piece and lock in, and they're going to find their way to where they're supposed to be, because this one might have A, T, C, C, G, T, and then that one has the matching, I don't remember what letters I just said, but the matching complementary DNA, so that they can stick together and bind. It'll be successfully implemented into the host DNA then, and they can make the corresponding protein. Yeah, so this could have major implications, like with designer babies, where we're going into, you know, developing embryos and changing the gender of the baby or changing the eye color, because we can cut out their gene for eye color and input a new gene for eye color, because we all transcribe and translate genes the same way. We can easily do that. So this is definitely a hot topic and a controversial one at that. But those are the main things about how CRISPR works. I would say that the majority of the question, though, is probably going to be asking you about like transcription and translation stuff, and why this is important with regards to that, or maybe they're going to make you analyze some data or something. Okay, so speaking of, then let's transition into talking a little bit about gene expression, just so that I can cover that as well, because that will probably also show up with this CRISPR question. So let's take a quick look at that. So I actually really like this diagram from Khan Academy, because it shows a little bit of everything that you need to know about eukaryotic gene expression. And it also blatantly at the top says eukaryotic gene expression, because this is different than prokaryotic gene expression. And I've seen that come up as a free response question before as well. I should try to find that so that I can show you. I will in a second when I'm done going through this. Okay, so a couple things to know about eukaryotic gene expression. You'll notice at the top we have DNA wrapped around. So we have chromatin, which is really condensed DNA, right? It's wrapped around these proteins called histones, which are what you can see right over here. And that's actually, I mean, this is like high level, but since we're talking about it, that's actually a level of gene regulation that we have. Because we can't go ahead and transcribe this DNA if it's actively wrapped around a histone, right? So right now this would be showing us like replicating this region of DNA that's unwrapped, but the regions that we unwrap versus wrap actually determine which genes are going to be transcribed at any one time. So that's, you know, just probably a stretch point, but that could show up maybe. So first step, we have transcription. So RNA polymerase is coming along and reading the DNA and making a complementary mRNA strand. Technically, that would be pre-mRNA, and you can see that it's like light purple and dark purple mixed, and we'll talk about why in a second. Remember that mRNA is single-stranded and has uracil instead of thymine, also has ribose instead of deoxyribose as the sugar group there. So then the part that's different between eukaryotes and prokaryotes is this processing region. So in this region, you can see we're actually removing the dark purple sections of this mRNA. Those would be the introns. They stay in the nucleus. And then the exons would be the light purple DNA. Those are the one, or RNA, sorry. And those are the ones that are actually able to leave the nucleus and become the part of the coding message. Now, so this is called splicing. We're moving introns and we're keeping in exons. I just saw on the practice exam from this year a really interesting question about this that asked students if there was a mutation in an intron region of this mRNA, what would happen to the resulting protein? And it was a trick question. The answer is nothing, because introns are removed. They don't code for proteins. Now, an intron is not always an intron, so we could actually make a different protein from this DNA sequence if we just cut out different parts of the RNA. So we could cut out the light purple parts the next time and make an entirely different protein, and that's how we can actually like 100,000 proteins from about 20,000 genes, because we can remove different sections and then we have a different end product. So that happens, and then you'll also notice we have a 5' cap and a 3' poly A tail here. The 5' cap is going to guide us to the ribosome, and the 3' poly A tail is going to protect us from degradation. There are enzymes here that are trying to snip away at this single-stranded nucleic acid, because viral DNA or RNA is often single-stranded. And so in order to protect our message, we have this like basically buffer or bumper at the end. If you snip off those poly A's, it doesn't matter. They're not part of the coding region. Okay, so that's transcription in a nutshell. Again, that doesn't happen. So the spliceosome is the, oh, sorry, there's questions here. Let me just make sure that I've answered all of them. What determines what are introns and what are exons? So the spliceosome is the enzyme or the protein that's actually responsible for that. And again, what's an intron is not always an intron. And you don't need to understand the total intricacies behind that. Just know that the spliceosome is in charge of that. Introns are removed. They stay in the nucleus. Exons stay as the message, and they get to exit the nucleus. So it's a little intuitive or counterintuitive, because you want to think that introns stay in the message, but they don't. So that is true. Spliceosomes tell it where to cut. Yeah, okay, all of you are right. Sorry, just I realized I was on my actual PowerPoint and not looking at your questions. I also appreciate that you're asking the questions in the chat as opposed to asking another question in this one, because I can't see those as easily. So thank you for that, and thank you for responding to one another as well. Okay. In prokaryotes, none of that processing process happens. So they don't shorten their mRNA, and they don't add the 5' cap and 3' poly A tail. So let me really quick try to find intron, exon, biology. Let me see if I can find one. I think I found it. The internet is a wonderful thing. I think it's from 2016. I think it was question 4, if I'm remembering correctly. That's like sticking out in my mind. Sure was. Look at that. All right, question 4 from 2016. So I'm going to post this in here in case you just want to real quick take a peek. So the question states, predict the length of the mature gene mRNA if the full-length gene is introduced and expressed in prokaryotic cells. And it tells you that the transcript is 15 kilobases long, but it says in eukaryotes, the mature mRNA is 7 kilobases in length. So for prokaryotes, you would need to say that it would actually stay as the 15 kilobases in length, because prokaryotes do not remove introns. They do not go through this process. So you needed to know that. You needed to know that while this mRNA gets a lot shorter in a eukaryote, because we're removing sections, prokaryotes do not do that. And so therefore, the length of the gene from the start is going to be the length of the mRNA at the end, because they do not process their mRNA. You can see the answer key there. Exactly. So that one's going to stay the same. Cool. So that's one big difference. Okay. So then translation is really, really straightforward. So our mRNA binds, it's read in 3 base pairs called codons. An anticodon on a tRNA molecule matches up with it. And depending, so like the easy example is AUG, because that's always our starting codon. It codes for methionine. So if a mRNA says AUG, the corresponding tRNA would be UGA. There we go, my brain. So AUG, UGA would pair with that, and then that specific tRNA molecule would drop off methionine. And that methionine amino acid would be the first amino acid in a polypeptide chain, which is just another fancy word for a chain of amino acids that will turn into protein. So that's going to continually happen. That's where your codon chart comes in. Translation is, in my mind, a lot more straightforward than transcription. So all the amino acids get added. A peptide bond forms between them, which you can see here as like this string, if this was like a necklace. And then finally, it will get activated, snipped, processed, whatever needs to happen to it in order to make it an active protein. And then it folds up into its 3D shape. Because the power of the protein comes from its 3D shape. That's how like different, especially like enzymes, different things fit into that 3D shape. And so that's an important aspect of protein formation. Something to note also with that is that because proteins gain a lot of their power and ability from their 3D shape, they can easily be denatured. Heat, pH, salt, things like that. If you've ever done an enzyme lab, you've probably manipulated one or all of those variables and seen that very quickly that enzyme becomes inactive because it's no longer able to possess its 3D shape, and therefore those bonds start to break, sometimes forever, sometimes just temporarily, but again, the protein is no longer active if it loses that shape. Okay. All right, cool. Wow, so we've already gotten through a good amount of questions. Looks like we have a few left. Okay. Endocrine system, what should you know? Great question. So more importantly than anything, you should just understand signal transduction, which we've talked about a good amount. We went over yesterday as well. So let's see. Let me pull up really quick on PowerPoint. But again, cell signaling, like cell communication, that's really going to be the big one. I would be familiar with like insulin and glucagon because those are really important enzymes that get talked about, or sorry, hormones that get talked about, not enzymes. And you need to know like positive and negative feedback. So let me pull up my endocrine system PowerPoint really quick. I'll share it with you guys. We can zoom through the parts that I think are most important, and then we'll jump into a little bit with energy efficiency. Water potential honestly usually doesn't show up. It's sometimes one grid in question, but I personally don't teach it to my students because I don't think it is worth the time that it takes to learn it. Because again, one grid in question is certainly not worth the struggle. There are other things that we could focus on. So I would not stress about water potential. Also, it's just an equation, so you can honestly figure it out on the fly if you need to, but I don't think it's worth your time, personally. Okay, endocrine. Sorry, it's taking forever for this to load. Are you guys seeing this? Okay, perfect. Oh, Gibbs free energy. Yeah, you don't need to worry about that as well. If anything, it would be one grid in question, and there's an equation for it on the equation sheet. It's in the bottom right-hand corner. So if you're in a pinch, you can use that, but it's not going to be like a free response question or anything like that. Okay. So endocrine, series of glands that are regulating activity. They produce hormones. Hormones are long-distance signaling molecules. Some of them are steroid hormones, which means they don't need a receptor. Some of them are not, which means they do need a receptor, and so that they only target certain cells. Negative and positive feedback, the difference here is just if the stimulus is inhibited or if the stimulus is enhanced. So with negative feedback, the stimulus is inhibited or decreased. That's what the response does. So I'll give you some examples of that. And with positive feedback, the stimulus is increased by the response. And I can show you an example of that as well. So some negative feedback examples: temperature regulation, blood glucose levels, and water limitations. We'll go through temperature regulation and blood glucose really quickly. So temperature regulation, so negative feedback, and this is all part of endocrine because it's hormones that are signaling these changes, especially with blood glucose, which we'll talk about in a second because that's an important one. With negative feedback, you're just trying to maintain homeostasis. You're trying to maintain a regular level. So when your body temperature falls, we bring the temperature back up. And you can see the examples of how we do that there. When your body temperature rises, we bring that temperature back down. Sometimes my students get confused because they think our body temperature is increasing, so therefore it's positive feedback because we're increasing. It's not, that's not true. The stimulus is that body temperature fell, right? So we're not increasing the stimulus, right? The body is not becoming colder, right? That would be positive feedback if our body got cold and then continued to get colder. But because our body gets cold and we reverse that stimulus, we decrease that stimulus by bringing our temperature back up, it's negative feedback. So you always want to think of this in comparison to the stimulus, and if the stimulus is being increased or decreased. If the stimulus is being decreased, it's negative feedback. And again, anytime you're trying to maintain homeostasis, so if we go too high, we come back down. If we go too low, we come back up. That's going to be negative feedback. The most important example of this in my mind is blood sugar levels. So you should really be familiar with insulin, glucagon, glycogen. Spelling does not matter on the AP exam, but it does matter for things like glucagon and glycogen, because if you spell it wrong, they're not sure you know what you're talking about, unfortunately. So yeah, if you could spell insulin wrong and it wouldn't matter because they know you're talking about insulin and they know you know what that means. But if you spell glucagon or glycogen wrong, they might not know that you know what you're talking about. So here's how this loop works. So as our blood glucose levels increase, pancreas is going to stimulate insulin production, and insulin is going to target body cells like tissues to incorporate glucose into them, to take in glucose from the blood. It's also going to stimulate the liver to produce glycogen, which is just like a combination of glucose molecules put together. And then because we're storing in body cells and we're storing in the liver, our blood glucose levels are going to decrease. Okay. On the same vein, when our blood glucose levels go down, the pancreas is going to release a hormone called glucagon, and glucagon is going to do the absolute opposite thing. It's going to have body cells release glucose, and it's also going to have glycogen being broken down in the liver into glucose so that our blood glucose levels rise back to normal. Remember, we need a specific level of glucose in the blood at all times because we use that for cellular respiration, and so therefore we really need to have that available to all of our cells at all times. All right. Okay. Positive feedback. Okay, so onset of labor, lactation, fruit ripening, and clotting. Again, here the stimulus is increased. So childbirth is a perfect example of this. We have contractions. Contractions are going to release a hormone called prostaglandins, which is going to lead to more oxytocin being released. Then the brain is going to release even more oxytocin. Oxytocin is going to lead to more contractions. So you can see the oxytocin levels are going to be continually increasing in the mother until the stimulus is removed, aka the baby is born. Okay, but those levels are not going to go down unless the stimulus is removed. They're increasing, 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 increasing. So more of the thing makes more of the thing makes more of the thing. That's positive feedback, and this is also endocrine example. Another example is breastfeeding. As the baby is sucking on the breast and milk is coming out, it's going to stimulate more milk coming out as well as more milk being produced. So that's an example of positive feedback through prolactin and oxytocin, two hormones that are going to lead to more milk and more milk coming out and more milk being produced. So those are good examples of like high level endocrine stuff. And again, cell signaling goes along with that as well. I have a few other examples in here. Did I already post this? I think I did. But here is really how this hormone would actually send a signal. So I like this one because on the left-hand side, we have a steroid hormone, estradiol. And you can tell that it's a steroid hormone because it comes right through the plasma membrane. It does not need a receptor on the outside. It has a receptor on the inside. And when it meets up with that receptor, we go through transcription and then translation to make a target protein. So that's just one way that this can happen. Obviously, we know about ligands and receptors as well, and those are for things that aren't steroids or lipids. Like you can see over here, this insulin receptor, that's going to stay on the outside and lead to a signal transduction pathway. Okay, cool. All right, we're moving, we're grooving. All right, a 2015 FRQ about energy efficiency. Ooh, I gotta look this up real quick. Okay, 2015 free response. Let me pull it up. I'll post it in here as well so that, oops, I don't need to be looking at an AP calculus free response. I do love AP calculus. Okay, so here's the free response. We're going to try to find the one. Oh, the endocrine, did I not send that? I thought I did. Oops. Here. Sorry, guys. What's going on here? Gosh darn it. Ah, why is this happening? My computer is not in good shape right now. Okay. There you go. Okay, great. Sorry, 2015. Did I post that yet? I can't remember. All right, I'm losing my mind. I'm sure everyone is. Okay. So there is your 2015 free response that someone was asking about. Oh, shoot. Okay, energy efficiency. Let's see. So it's not the first one because I know that one's about circadian rhythms. It's not the second one because that's about, okay, not that one. Daughter cells. Energy efficiency. Can anyone who asked the energy efficiency question tell me which question in 2015 they're talking about? Because I do not see an energy efficiency one. Oh, is it the math question maybe? Of question 2, part C. Maybe that's what you're talking about. Yeah, probably that's what you're talking about. Okay, so we can go through this. If you guys look at question 2, part C, that's on page 6 of the thing I just sent you guys. Wait, I can pull this up on the screen. Why am I doing this? Why am I overly complicating? 2C, thank you. Okay. Actually, let's just pull up the answers and talk through. Because these are actually really easy points. If you got it right, these were really easy 3 points. I think it was 3 points. You guys know that you can figure out how many points everything is by how many bolded words there are. That's like a telltale sign. So that's helpful. You can see how many points you're getting for each part of each question. The only time that that's not true is sometimes there's a bolded word, but it says and justify both claims. So then justify that would count as 2 points, not just 1. So let me pull this up. Okay, scoring guidelines. Cool. All right, so let's read through this part really quickly together. So it says, a researcher estimates in a certain organism the complete metabolism of glucose produces 30 molecules of ATP for each molecule of glucose. The energy released from the total oxidation of glucose under standard conditions is 686 kilocalories per mole. The energy released from the hydrolysis of ADP to ATP under inorganic phosphate under standard conditions is 7.13. Calculate the amount of energy available from the hydrolysis of 30 moles of ATP. So if each mole of ATP or molecule of ATP can produce 7.3 kilocalories per mole of energy, then all you need to do here is do 7.3 times 30. And that gets you your 219 for calculate energy available in ATP. Okay, so again, all I did there was the 7.3 kilocalories per mole times 30 moles. So this is really a chemistry question. And then you get the 219 for that first point. So then the second question says, calculate the efficiency of total ATP production from 1 mole of glucose. So they're telling you that burning glucose is 686 kilocalories per mole, but we're only making 30 ATP and getting 219 kilocalories per mole. So we need to do 219 divided by 686, which would be 100% of the energy. Okay, or you can set up a proportion there, but if you just do 219 divided by 686, which is the total energy we should be getting, you get that 0.319 or 31 or 32% efficiency, okay, because we're getting 30 ATP, moles of ATP. I divided by 686 because that's the total amount of energy under standard conditions that's available from a molecule of glucose. And that's given in the problem. Second sentence, the energy released from the total oxidation of glucose under standard conditions is 686 kilocalories per mole. But we're not getting all of that energy. We're just making 30 ATP from this. Okay, we're not burning glucose like you would in the conditions under a lab, which is what they're talking about. So we get 7.3 kilocalories per mole. So if we make 30 moles of ATP, 7.3 kilocalories each, 219 kilocalories of energy per mole divided by 686, which is the total that we could have gotten, and we're about 32% efficient there. What happens to the excess energy? Anytime we're losing energy, we're not actually losing energy, we're just releasing it as heat. So you can truly always say that. Yeah, heat. That's also why we lose so much energy. They do make it sound so confusing. You guys are so right. It's really crazy because it's actually really, really simple. And I overthink them too. So don't feel like you're alone. And my students do too. But it's never as hard as you think it is. Yeah, I think the wording of this was hard. What I would also say, so really quickly, let's see, average points AP Biology 2015. Let me see if I can find this quickly. If I can't, it's not a big deal. But the average points on these long free response, just so you guys know, is usually like 3 points. So let's see. I think I actually found it. Okay, this is great, guys. Ready for this? You'll love this. Okay, the average amount of points, and remember, average is going to be like a 3 on the test, is a 2.89 out of 10. A 2.89 out of 10. So think about how many people messed this question up. And if you look at the other points of this, this question, there are a lot of easy points there. So what I would say is, and this is why I always tell you guys to do the short free response first, like think about how much time and angst would have gone into you trying to tackle this problem. You could easily spend, if you look at this problem, this question too, you could easily spend 45 minutes on this question. There's like no doubt. And 45 minutes is half of your free response time, and there are a lot easier questions. So actually, if you look through 2015, I actually really like this year because it's got a lot of really cool stuff. So like this is these are easy points, this cladogram. Let's see, there are other really easy points in here. Like question 8 is super straightforward. It's just about like B cells and T cells. So for whoever asked about that, we can actually talk about that next. Yeah, I don't know. There's a lot of easier points here that you could have gotten. So try not to stress so much. So is it always that low? Not always. Sometimes there's like four points. I've seen 4 points. That's a great question about the plus members power points. I'll get back to you. But yeah, it's like an 80% to get a 5. College Board, yeah, they're mean. They're really mean. They're out to ruin your life. Duh. But we just got to be better than them. Okay. All right. So that's that. Okay. Hide. Perfect. Answered that question. Hey, we're cruising today, y'all. I'm proud. Okay. Perfect. It was the next question. How detailed do we need to know about the immune system? Okay. So you should know like non-specific versus specific response. You should know like the acquired immune system. You should know that like B cells produce antibodies and T cells deal with infected cells. But let's take a look at question 8 from this year because I really like this one. And this will tell you kind of like what they were expecting of students this year as far as B and T cells go. Okay. You would need to write that in a sentence for that. Sorry, that math would need to be in a sentence. Yeah, okay. And pictures, if you're drawing a picture, you always need to describe it in words. They're not going to grade your picture. So just some things to keep in mind. Not that a picture can't help, but then you need to fully describe it in words as well. Okay, so this question from 2016, an individual has lost the ability to activate B cells and mount a humoral immune response. Part A, propose one direct consequence of the loss of B cell activity. So this question is just saying, how do B cells impact the initial exposure? Okay. And then the second says, how is this going to affect second exposure? You need to know that B cells have memory, that B cells produce antibodies. So for part A, if we lose B cells, how is that going to affect our first exposure? Well, we're not going to produce antibodies and we're not going to produce memory, but you only need to say one of those things. So if you know that B cells produce antibodies, you can just say that, oh, we don't have antibodies because that's what they produce and now we don't have them anymore. Part B, it says propose one direct consequence of the loss of B cell activity on the speed of the second exposure. You would need to say that it would be like the same as the first, or that it's not faster than the first, or that it's more slow than we would think from a second response because we don't have memory. So if we don't have memory, then the second exposure is essentially the same as the first exposure. And then part C, describe one characteristic of the individual's immune response to the bacterial pathogen that is not affected. So here's where you just needed to know one other thing about the immune system. You could say T cells, you could say cell-mediated immunity, you could say non-specific immunity, you could mention truly any other part, even skin, they have epidermis down there. All of those things would not be affected if we lose B cells. So that's kind of the extent of what you would need to know about the immune system, a little bit about B cells, a little bit about T cells, and a little bit about the non-specific response. You should also know that because we form memory with B cells, like a secondary exposure, you could talk about vaccines, like your primary exposure is with that weakened or killed virus. Then the second time you see it, you mount a stronger response and you don't even know that you've been affected by it. Okay. Cool. So for part A, do you have to explain why? You don't need to explain why. No, you don't need to explain why at all. Or you can just say we won't have antibodies because B cells won't produce them. If you say that, that's kind of implied in the question. Cool. Predictions for FRQs, that's a great question. I personally think there's going to be a chi-square, and if you guys want to practice that, I went through it in, I think, last night's review session, maybe Wednesday's review session. Honestly, I cannot remember, but look through that. It's the free response from 2013, question 1. I think there's probably going to be something about self-communication. I don't know. I don't have any major predictions other than that, unfortunately, but you guys will know before me. Oh, last night. Thanks, guys. So last night, so you can rewatch last night's. So how can you keep your free responses short and direct? That's a great question. Well, what I would say is, if you don't know what you're talking about, don't over explain anything. So one way to just keep yourself short and succinct is to start with the short questions and then go to long, because you'll be in the mindset of writing shorter answers. I would say also give yourself a 1-page limit for the long free responses. I have never not been able to answer a long free response in less than a page. It's always less than a page. And if you don't know what you're talking about, you'll write more than that, because that's what we do when we don't know what we're talking about. I would also say to keep yourself accountable, skip around. For a long free response, there's usually 5 parts, so skip around and focus on the ones that you do know, because again, you're going to start rambling. If you know what you're talking about, you will be short and succinct and you won't have to think about it. It's just when you don't know what you're talking about. Math questions, you just need the answers. You don't need the work. So there's your answer to that one. Do all the bullet points need to be answered? No, it's just 1 point. So you just needed one of those 3 bullet points, or one of those 2 bullet points, or one of those 4 bullet points in order to get the points. You do not have to answer the questions in order. I would suggest that you don't, especially, right, if you're working from short to long, which again, I highly suggest you do. And then you get to, let's just look at an example, because this is really helpful. Let's say we get to question 1 here. And there's a graph that needs to be constructed. If you hate making graphs, then you should be answering, and you don't have time left, then you should be answering, oh wait, I'm on the wrong one. Sorry guys, we're looking at 2015. I was gonna say that's this question from 2015. Okay, so 2015, oh yeah, it's this crazy question about circadian rhythm. So I think this was a five-part question. The first part is about the nervous system. The second part is about the figure. So I would start with part B, because you literally just need to look at figure 2 and analyze it. And so you're going to get a point there without even having to think, especially if you're running out of time. Then I might move to part E, which is all about natural selection, because you can answer that question without having any idea of what's going on about anything else. And if that's all I have time for, at least I still got 3 points. And I think the average on this question was under 3 points. So I've already beat the average just by doing 2 really, really easy parts of this. And I've written 3 sentences. So you don't have to label A, B, C, D, E. You don't have to do that. I highly suggest that you do, because it just makes it easier. To make sure you don't label them wrong. If you label them wrong, it honestly doesn't matter. They will still give you points no matter what. You can answer part C and part A, and they'll still give you points for it as long as you're not contradicting something. But again, my suggestion is always to label, because it's going to make it easier for your reader to grade, and anything you can do to make your reader's life easier means that you, I don't know, you could get, you always want to keep your teachers happy. You always want to keep your graders happy. Okay. So Daniel, you would start at the back of the booklet if you're working from short to long. So you're going to start with question 8 and then move back to questions 1 and 2. But you also have that reading period where you can kind of flip through and see what the questions are like. I just would highly, highly, highly suggest that you work with questions 3 through 8 first before you look at questions 1 and 2, because questions 1 and 2 are time sucks, right? Even if they're not that hard, they take up a lot of time. So you would rather get the points that you can get on the easier, shorter questions, and then tackle those long questions knowing that you don't have to pace yourself anymore at whatever time you have left. Yeah, you can definitely start writing during the reading period. It's included in the 90 minutes. It is. Yeah, for the questions. Are there designated pages? Yes, there are designated pages. So they'll be question 1, and then it'll have 4 pages of lines. And that's also really intimidating that they do question 1 with 4 pages of lines and you don't need to answer in 4 pages of lines. Let me see if I can find an example, because I know there's lots of questions here. I'm trying to see if I can find an example. I think all the practice exams are the ones that have it set up for you, and I can't share those with you, unfortunately. But it would have the question, and then it'll have the lines right underneath that question. So it's not like a booklet. It's like a packet where the questions and the lines are connected, if that makes sense. So then you can definitely answer the questions in random order. So that's why I start at the back. So you'll have the question, and then you'll have the lines for question 8, and it'll say, yeah, exactly. Yeah, it's not like APUSH or any of those other classes. It says question number, and it's all kept together. It's not like questions are not separate from answer space, if that makes sense. Yeah, you can look at a student response, I guess. That would be a smart idea, which might be, yeah, here we go. Sample student scoring. Brilliant. There you go. So you can see right there. Additional page for answering question 1. Cool. Okay, let's see if there's any other quick questions I can answer. Okay, action potential. I've talked about this at great lengths. I'm actually not going to talk about it tonight. I talked about it on Wednesday and Friday, so you can rewatch either of those. Primary productivity, population growth calculations I also did last night or on Wednesday. I can't remember, but I definitely did a practice there. Yes, you can answer the individual parts of a question in random order as well. All the random things can happen. Are the labs tested on the exam? So labs could show up on the exam. Yeah, again, it is mainly in FRQs, but sometimes it's in those, if you remember, like doing a practice test where you have like an experiment set up, and then you'll have like 5 questions that follow. Sometimes they'll play on the labs in there too. They always give you all of the information that you could possibly need. You just need to analyze it. So you do not have to have done that lab before. I honestly wouldn't spend any time studying the labs. As long as you understand the concepts that the labs are testing, you will totally be able to answer the question. So don't feel like you have to come in with knowledge on all the labs. I mean, it's always helpful because you've done it before, but more importantly, you just need to be able to analyze data, and you need to understand the concepts behind the labs, if that makes sense. Yeah. Okay, so DNA is always going to be more specific than morphological data. I'm answering this next question. Morphology is how something looks. DNA is obviously what codes for those looks and appearances, and so morphology is much less specific. Although there was a question, and I can't remember what year, that asked this, and they would accept your answer either way as long as you defended yourself. So that happens a lot. It's just like, pick your poison. As long as you can justify it, they'll give you the points for it. But again, morphology is just the way something looks, and DNA is what actually codes for proteins. So it's going to be more specific because that's what's inherited from generation to generation. Yeah, so don't panic too much about the labs, but there is a lab review. Are kiddies on the exams? No, they're not, except for, yeah, talking about ADH, right, and excretion, but you don't need to know the excretory system. ADH also, they would provide you the information of what ADH does, and you would just need to be able to apply it. So quick tip on these guys, with any of these like hormone signaling situations, I always draw myself, while I'm reading the question, I draw myself arrows of how everything's connected. So if they tell me that ADH is going to increase water retention or increase blood water levels, like I would write that out so that I don't have to continue to reread the question, and I can see how everything's connected. So I always annotate questions about human body a lot, because they usually have different hormones and stuff involved. But you don't need to come in knowing what ADH does. They would tell you. Okay. Okay, so I also covered this last night with neurotransmitters, but basically calcium comes in at the end of an action potential. Calcium is brought into the end of the axon, which is going to stimulate the release of vesicles that have neurotransmitters in them. Then neurotransmitters are released. They go across the synapse, the synaptic cleft, which is the space in between axon and dendrite, and then those neurotransmitters would stimulate the action potential on the other side, usually by opening a ligand-gated sodium channel, which is allowing for the influx of sodium to come in, which is depolarizing the neuron. Okay. Yes, calcium channels are voltage gated, which is why they open up when the action potential comes through, when the neuron gets depolarized. Do exams get harder every year? No, it should be the same every year. But it just depends on what you studied. So sometimes it feels harder, sometimes it feels easy. It just depends on what they focus on that year and if you feel comfortable with that thing. Oh my gosh, yeah, I have that packet of grid-ins and I still haven't figured out how to upload that. Shoot. Attach a document here? That would be amazing. Oh no, none of that's helpful. No, you can't separate the pages. Okay, I'm gonna do something really quick, guys. Did I already share this PowerPoint with you? I believe I did. I'm gonna really quick snip in some images here of grid-in questions because you guys have asked a bunch and I have to give them to you. And let me make sure, can you guys just really, you have access to this, right? It looks like you do because I can see a lot of people on here. Okay, so I'm just gonna really quick snip. These are all practice grid-in questions. So while I'm snipping, I know I've already shared this with you, but with the grid-in questions, I always say to go to them first. I always think there's like 3 that are really easy, simple algebra. Then I think that there are 3 that are oftentimes really, really challenging. And those are not going to be worth your time. Remember, it's worth the same as a multiple choice question. So if you're spending 7 minutes on it, you shouldn't be. Remember that it equals out to, so it's 69 questions in 90 minutes. So it's about 1 minute and 20 seconds that you should be spending on each question. So if you feel like you're spending a lot longer than 1 minute on a question, you should be moving on because it's totally not worth it. I'm just realizing right now the one I'm about to add, question 5, is a Hardy, no, it's not a Hardy member. It's a chi-square problem. That would not be a grid-in, but it could be a free response. So don't panic. If there was a chi-square as a grid-in, you should skip that immediately because it takes way too long. So it's 150% not worth it. I'm also putting up right now when I'm with this question 6. Question 6, I would also be like, you need to skip that if this was on your AP exam because it's telling you to apply a brand new formula. And applying a brand new formula is not something that you should be wasting your time on an AP exam because it's going to take you longer than 1 minute and 20 seconds. This is just like a smattering of questions that ask about a whole bunch of different things. And you'll notice that, again, some of them are really challenging. And remember that just because you can answer something doesn't mean you should answer something, okay? I know we're all stubborn, we're all smart people, and we're all stubborn, but that does not mean that you need to spend the 8 minutes that it might take for you to answer question 7 that I just posted. Because it's not worth it. They're worth the same as a multiple choice question. But hopefully this is helpful, and my ramblings while I'm doing this is also helpful to you. Yeah, so here you guys go. Hopefully these help. Cool. All right. And that's like almost all of our time, but okay. Oh my gosh, yeah, no, for the grid-ins, please, for the love of God, don't spend 10 minutes. That's so much of your time. They're not worth any more than the multiple choice questions. So if you're spending more than 1 minute, you should honestly move on. Daniel, I kind of hope chi-square is long FRQ too. College Board might do that, and you're going to skip it because you're better than College Board. And so you know that it's not worth your time. Guys, I just have to remind you, like none of these, there's only 6 of them, and none of them are worth more than 1 minute, and none of them are worth more than a multiple choice question. So you have to like really, really, really work. The thing that I need you to do is just not get hung up on this stuff and be like, okay, again, I know I can answer this math problem, but it is not worth my time to figure it out right now, right? I'm gonna move on, okay? So find those 3 easy questions, get those done. Do not worry about the other ones that are really, really tricky. If you have time at the end of the multiple choice, yeah, give yourself a compliment. But like if you have time at the end, then what is the, what's the harm? Then you can go devote that time to figuring out that really hard math problem and you'll feel so good. But if you don't have time, you're going to be really mad at yourself in the end. So remember, multiple choice go from easy, medium, hard, medium, easy. So if you get really caught up in grid-ins and then you move back to the multiple choice and you get like stuck in the middle, think about all these easy points that you're missing at the end because you were too proud to move on in the beginning. And again, that goes the same for, a really, really hard or long multiple choice question as well. Yeah, you can have a calculator on the multiple choice and the free response. They basically any calculator is allowed, but the calculator policy is on College Board's website. As long as it can't connect to the internet, you're fine. It used to just be a 4-function calculator, so thank God that you guys don't have to do that anymore. In one sitting to get the widest scope, truly any of them. They're all relatively equivalent, Ria. So I would pick any of them. Yeah, like 2016 is a good year. 2015 I liked. 2015 was evolution heavy. 2016 was ecology heavy. 2017 is decent, actually. 2017 does a good job of going over all of them. You guys are way too kind, but I'm really glad that I can help you now. And come back tomorrow too, because we have another hour. Ecology, don't stress about. Let's talk about that tomorrow, because you should not be stressed about that. It's just graphs, guys. You can analyze a graph, I promise. Yeah, cool. You guys are great. Come back tomorrow. Last review session. It's 6 to 7 Central, so 4 to 5 Pacific, 7 to 8 Eastern. Had to get it done before Game of Thrones, obviously. But again, make sure you're getting some rest and getting some studying done, and I'll see you guys tomorrow. You're going to be great. Bye, guys.